Good afternoon, everyone. Good evening around the globe. Thank you for joining us today. I'm glad to announce that we have over 32 states represented. We have folks from South Korea and Ireland and Finland and Egypt and Paraguay. So thank you for joining us from around the globe, whatever time it is. My name is Brian Lewis. I'm the president of KidneyCAN, and we're really pleased to have you all here today. We're also, you can find out more about us at kidneycan.org. We're thrilled to have two world-renowned oncologists, medical experts in kidney cancer with us today. But before we get started, I've got to go over a few housekeeping notes, so just bear with me for a moment. First of all, we're streaming live on Facebook. If somehow you get disconnected, you can pick it up over there. We've got a chat feature here today. Please use it if you'd like to. And it's also, this is going to be recorded and posted on YouTube, so you can come back and watch it some other time. The second disclaimer here is that there are going to be a lot of insights today and advice, but KidneyCAN's not a medical organization, so we can't be dispensing advice. So please consult with your own healthcare team on any of today's topics. Thirdly, this is a town hall, and so we're asking you to pose your questions on behalf of the town, the community, the kidney cancer community. So we might have to cut off your question if you get too into the details about your diagnosis, if that's what you're asking about. So please bear with us there. Finally, we're going to try out a polling feature today, about a 30 to 45 second response time. So if you would join us in the polls, that'd be great. We're not going to be sharing any of this data. It will be anonymous. At the end, we might ask for your information if you would like to join with us. So with that, let's call this town hall to order. A quick overview. We're going to reflect back on 2020, and then we're going to look forward at 2021. So I'm really pleased to have our panelists with us today. I hope, hopefully we can see Dr. Choueiri here and Dr. Uzzo coming up. If you haven't met Dr. Choueiri, I sometimes think of him as a former star midfielder for a European soccer team, but that's after we retire and we find a cure for kidney cancer. But now he's the distinguished chair in oncology at Dana-Farber Cancer Institute. He's also the director of the Lank Center for GU Oncology, and he's a professor at Harvard Medical School. Dr. Choueiri, pleased to have you. Thanks for joining us today.
Thank you, Brian, and welcome everyone.
Thank you, thank you. Also, Dr. Robert Uzzo is here in Philadelphia. Quick side note, I heard that you've been to Antarctica. I want to hear about it sometime. I'm looking forward to someday going there. He's at the world-renowned Fox Chase Cancer Center. He's a urologic surgical oncologist. He's also the chair of the Department of Surgical Oncology. Both of them have in common Cleveland Clinic in their backgrounds, and I think there's a Cleveland Brown fan that's going to talk to us in a moment, but we'll see. So Dr. Choueiri, kick us off here. Tell us about some reflections on 2020, and then we'll hear from Dr. Uzzo.
Yeah, so thank you for having me here, and welcome everyone from the 32 countries, and hopefully next time will be 132 countries. And glad to be talking about kidney cancer in 2020. I don't know, when I think about 2020, what to think about that year beside COVID and the pandemic that, you know, turned our lives upside down. But let's just, you know, discuss kidney cancer. In 2020, we continued to push the envelope. There has been new press releases, a new presentation that came in patient with metastatic disease. As you know, patient could still present with metastatic disease, or their disease can come back after surgery. And we have combinations of immune checkpoint inhibitor immunotherapy and target treatment with targeting the VEGF receptor and other receptors that showed significant activity and efficacy against single agent target treatment. So drugs like sunitinib, which were drugs that are the standard, are not anymore standard. So this is not new. This follows on data from 2018, 19, but we have more and more mounting data. One in particular combination is cabozantinib and nivolumab that was presented during the virtual European Society of Medical Oncology meeting in virtually, obviously, in the fall. The other one is a press release of a combination of pembrolizumab, it's a PD-1 inhibitor, and lenvatinib that showed also to beat sunitinib again. So the time where we use in a new patient single agent is really rare. So that was something, interesting to see, not unexpected. Also, two studies finish accrual in the adjuvant setting, meaning in patients that have kidney cancer. The kidney cancer is resected. There is nothing on the scans, but we know they're at higher risk for the cancer to come back because they have potentially what we call micrometastatic disease. The other thing that we mentioned is a lot of basic science and translational science research looking at potential biomarkers for kidney cancer treatment. You have treatment A, hopefully treatment A works in patient B, let's say. And we came closer to more and more clues. And, it's unfortunate a bit that we ask questions, we end up with more, questions after we answer these questions. So there has been some clues why mutation, for example, in PBRM1 could make the tumor more sensitive potentially to immune checkpoint blocker, but this doesn't happen all the time in multiple settings. There has been also some very nice work actually from colleagues of ours to detect kidney cancer at a very, very low level in the blood. This is not ready for prime time, but I think the liquid biopsies that you hear about, which has been very challenging in kidney cancer because it's a tumor that does not shed a lot of cancerous DNA in the blood. But now with novel and new technologies, we are able, to detect it. And hopefully in the next couple of years, we will be able to have more data about that, but we may have some tests that are coming. I think when I look at 2020, beside the pandemic, I see also how the survival and the median survival continues to improve in kidney cancer. Really, I finished my training in Cleveland. So both Dr. Uzzo and I, part of our training was in the renowned Cleveland Clinic. When we used to practice in 2005, 2006, you would see patients, the kidney are removed, the cancer came back, and some of the studies would randomize the patient to toxic treatment that did not work much, interferon, or sometimes to placebo, and the median survival used to be a year, less than a year. This is unheard of. We're pushing the envelope really, 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 quite high. The median survival, 4 years or more, at least on trial, of course, when we look at real world evidence and a patient that are sicker, sometimes it's lower, but I think there is a lot of room to hope. I don't know if I should say this is 2020 or I would say the end of 2019, if we can include, start from October 2019. One of the things that I would say kidney cancer was honored in a way that my very close colleague, you can see in the back here, although I don't think there's a focus, Dr. Bill Kalin, who's been a friend and a mentor, have shared the Nobel Prize in Physiology or Medicine in October 2019, based on his basic work on oxygen sensing and hypoxia, coming to this from an oncological, cancerous, if you want, angle. He shared it with two other scientists, one from Hopkins and one from the United Kingdom, that came through the same story from different angle. Why I'm linking this to kidney cancer, as you know, kidney cancer, the most common form of kidney cancer, have an alteration, a mutation in a gene called the VHL gene, which lead to downstream effect leading to, if you want, the increase in hypoxia and increase in these factors that drive the growth of blood vessel, including a central regulator of that processes called Hif, hypoxia-inducible factor, just call it Hif. So he had, worked and all that, and he was also very much involved in the whole process. And we do have now agents that we're taking to clinic that can target this Hif, this central regulator of response that was thought in the past, this is a kind of a factor, we call them a transcription factor, that are hard to target by modern drugs. But I think some elegant work at UT Southwestern showed that it's possible. So we have now drugs in clinical trial that are tolerated and they're making it to the next stage. So, I'm gonna stop at that, Brian. I think, and to mention that the main message here is to stay hopeful, that there are a lot of things happening in kidney cancer with you, Brian, and the staff at KidneyCAN in 2019, late 2019, something in me want to go even a couple of months before the pandemic started. And, we have started the first translational science meeting dedicated to kidney cancer research. Dr. Uzzo, of course, was a big part of it. And that's because the work of KidneyCAN and other advocacies for which led to the Department of Defense allocating funds specific to kidney cancer. So our seasoned as well as our junior scientists could apply and focus on kidney cancer, because we need all of us together, patients, clinicians, as well as basic scientists and families, to be all together focusing and channeling our energies into discoveries so we can cure kidney cancer forever.
Great. Thank you, Tony. That's an excellent recap of 2020. We're going to come back to you in just a minute. Let's turn it over right now to Dr. Uzzo, and then I'm going to ask Tony if you come back at the end of Dr. Uzzo's comments, and let's hear let's look across the horizon and tell us what we might see, Dr. Uzzo, coming up in the next 6, 12, year and a half.
Well, thank you. Again, my name is Robert Yuzo. I'm here in Philadelphia. I'm very honored to be here with Tony, a good colleague and friend. I did finish in Cleveland in 2000, so I'm about halfway through my career, and kidney cancer survival is 5 times longer than it was when I started my career. There are very few physicians that see that kind of progress in half of a career. Certainly it was unheard of in the cancer field. And so I came to Philadelphia, and we have a long history of basic researchers looking at kidney cancer here at Fox Chase. Al Knudsen, who was the person that first postulated tumor suppressor genes, won the Lasker Award, which is the American Nobel, and the Kyoto Prize. In addition, Ernie Rose won the Nobel Prize for ubiquitin-mediated degradation, which is an important way that the VHL protein is moderated in the cell. So like Tony, we have a long history of translational science. Now, the most important thing I can tell you is that the field of kidney cancer is not only changing with systemic therapies, but also it has changed dramatically in the localized setting. Let me give you some examples. When people get scanned and they're told they've got a mass on their kidney, doctors use the word mass or tumor because they can't say it's cancer. It's just simply a lump, a neoplasia, if you will. We're getting much better at using scans in a field called radiomics to tell us if those tumors are cancer, and hopefully therefore trying to avoid biopsy. I think what you'll see in the next couple of years is the integration of artificial intelligence and neural networks to make scanning more accurate, so that we're better able to say if a small spot is cancer or if that small spot is nothing. we can't do that yet. And so we rely on a lot of biopsies. So one thing I think you could look forward to in the next year is the use of artificial intelligence, neural networks, and radiomics in imaging. The second thing I think you could look forward to, and Toni alluded to this, is the field of biomarkers. Now, we'll talk a little bit more about this in the future, but I want you to hear there are really three different types of biomarkers. There are tissue biomarkers. Take a piece of the tumor, check the tumor. There are blood biomarkers, circulating DNA, circulating tumor cells. And then there are body fluid biomarkers, urine, saliva, other body fluids. All of these areas are being very aggressively studied. And I think this will be a real tectonic shift if we could correlate the biomarkers with the prognosis. So I think that's something that we'll talk about more, but you're asking me to look in the future. That will be a major next step in the evolution of kidney cancer. Now, in terms of surgery, we're using a lot more biopsies. In the past, 20 years ago, if you had a mass in your kidney, it was simply removed. We're using a lot more biopsies because we're realizing that not all kidney cancers are equally aggressive. And we're starting to understand the genetic differences between something like a chromophobe cancer, a papillary cancer, and a clear cell cancer. And even within clear cell cancer, this very vast heterogeneous microenvironment. I like you to start to think about cancer like sort of the bacteria in a pond, right? There's this evolution within that pond, the good bacteria, the bad bacteria, something comes along, you get a little bit of acidity in that bacteria, some will die, some will live, some will thrive, some will go away. The same kind of, we call it clonal evolution, occurs in kidney cancers. And ultimately, many people will have good clones, if you will, and other people will have bad clones. And we're trying to figure out what kind of clones will respond to what type of therapies, and then how to sort of reshift that environment, that microenvironment. And you're going to see a lot more work being done with tumor biopsies and circulating DNA. I'm asked again about the future. I think you're going to see a lot more ablative therapy, ablation being cryoablation, microwave ablation, and radiation therapy, targeted radiation therapy like Sbrt or CyberKnife to small renal masses. We're starting to see phase 2 and phase 3 studies in this. I'll tell you, in terms of surgery, there's going to be a new generation of robots. And so what we're really pushing for is outpatient surgery. So many of the people here have already experienced surgery for their kidney cancer, but you probably stayed a day or 2 or 3 or 5 for that kidney cancer. Some of you had the whole kidney removed. Some of you had pieces removed. Some of you may have had robotics, but I bet most of you didn't. But we're going to see a whole new generation of robotic surgery. And I think on the surgical side, we're going to see a lot more outpatient surgery in the next year or 2 for kidney cancer. Now, I'd like to talk a little bit briefly about adjuvant therapy, because you're asking me about the future. Right now, many of you know that when your kidney is removed or a part of it's removed, if you've got a high-risk kidney cancer, you just have to sort of wait it out and see whether or not it's going to come back. And this whole concept of microscopic metastases really breeds fear into patients, because they don't know, am I the person that's got a circulating microscopic tumor cell? And if so, can't we get rid of it somehow? Well, plenty of studies have been done. Unfortunately, most of them have been negative, with the exception of the Sbrt study, which most of you know led to the approval of sunitinib in the adjuvant setting. That is to say, after all the tumor has been surgically removed, you get a year's worth of this therapy. But frankly, it's not a home run. It's probably best a single at best, because it doesn't increase people's lives, and it really does have a lot of toxicities. So now we've put all these immunotherapies into the adjuvant setting, and these trials are going to mature, and they're going to start to read out in the next 18 months or so. And we're going to see whether immunotherapies prevent the recurrence of kidney cancer after surgery. That's going to be a major shift. I'll also say that this new field of consolidative surgery is going to be something you'll see in the next year. We're doing it already, But consolidative therapy means that after Dr. Choueiri gives you his magic medicines and shrinks the tumor down, can we then go in and surgically consolidate the results by removing what's left, right? It's kind of using my analogy, right? I've got this pond full of really bad bacteria. Dr. Choueiri can kill most of them. Can I come in and drain the pond? You know, hopefully that combination, that 1-2 punch, immunotherapy, targeted therapy, then surgery is going to continue to allow us to have people that are off therapy for, you know, months, if not years, before they recur, or hopefully not even recur at all. So keep your eye out on more consolidative therapies. And lastly, now this is not my field, but you're asking me to look forward. You know, this is far more for Dr. Choueiri to mention, but there are all sorts of new targets, you know, for kidney cancer. There are these metabolomic type of drugs now, these glutaminase inhibitors that people are testing to try to understand how to change the metabolism of kidney cancer cells. There are all these new combination of immunotherapies. There are macrophage therapies, new immunotherapies targeting macrophages or regulatory T cells that are sort of coming up. There are new therapies that are looking at stromal cells. But, you know, ultimately, the holy grail here is if we can find a tumor-specific protein that's only expressed on these kidney tumors, and Dr. Choueiri and his partners can start giving targeted CAR T cells, those chimeric T cell antigens, hopefully someday, to cure kidney cancer. So that's what I see. Changes in diagnostics, changes in surgery, and all these new therapies coming down the road in clinical trials, looking at different mechanisms to get rid of that swamp, if you will. I think you're muted.
Sorry, thank you so much. That was quite exhaustive. Really appreciate the passion there. Dr. Choueiri, do you have anything to add on to that wonderful summary?
No, I mean, this is like a tour de force review, but I think that the goal here, hopefully, is to remain very, very hopeful of the things that are coming and the community that are coming, you know, together for kidney cancer, you know, research and making kidney cancer outcomes better and better, from detecting kidney cancer by screening to curing patients, hopefully at 100% when we remove the tumor with the least amount of harm and preserving their kidney functions, even if they have another kidney, to hopefully curing patients that present with harder, you know, to treat tumors, especially when the cancer has metastasized and has spread, to focusing on tumors, like Dr. Uzzo was saying, that are not the conventional type of kidney cancer, that are grouped all under non-clear cell kidney cancer, if you want, non-conventional, which are, we're starting now to get them a bit by bit. What do they mean and what treatments to do and how aggressive to be or less aggressive about them. So I think the future remains and is today and will stay, you know, bright. And, you know, we're here to help and to move things forward, because at the end of the day, Dr. Uzzo and I want to retire and think about a different career. I don't know when, but, you know, at some point.
All right. Yeah, put KidneyCAN out of business while you're at it. Why don't you solve this? Let me ask a quick question. I think a lot of people, and we'll try to maybe circle back to this, but biomarkers is always referred to as the holy grail. Dr. Uzzo, you mentioned that there's 3 different areas. I know, Dr. Choueiri, you've done some work here and your folks up there in Boston. Can you give any, like, the next layer of where you think might have the best opportunity for breakthroughs?
I could try to start on that one. So the most rich area for this is to take a clinical trial and use the outcomes of that clinical trial to try to associate it with a biomarker. And this was recently done. Recently in Cancer Cell, I think, Bob Motzer and Brian Rainey and others looked at the Emotion 151 trial, which is atezolizumab, you know, plus bevacizumab versus sunitinib. And they took all these patients and they tried to aggregate these patients to see who responded best to one therapy versus the other therapy. Now, if we really wanted to get into a discussion about biomarkers, you have to ask, what question are you asking? Is it to diagnose patients with kidney cancer? Is it to tell me about their prognosis? Or is it to tell me about who's going to respond to what therapy? So biomarkers is a generic term, but biomarkers can be used for lots of different things. Now, what they did is they looked to see what biomarkers might say who would respond to therapy X versus therapy Y. And we're starting to see themes emerge. Now, most of these biomarkers now are in tissue. So you take a piece of the tissue, you study the genetics of the tissue. Dr. Choueiri mentioned before, PBRM1 seems to be a biomarker that has a better prognosis. All right. TP53 or P53 tends to be a biomarker that has a worse prognosis. And they've listed in their study, and others like it, these whole list of sort of when they test the genetics in the tumor, now what type of those mutations are more responsive to different types of therapies. So we can go through lists of them, and we have biomarkers for benign tumors and chromophobe carcinomas, and all this stuff is starting to emerge. But I think right now the best data is in tissue-based biomarkers. Hopefully this will then give us information for blood-based biomarkers. Tony?
Yeah, no, I agree. I think biomarker is a generic, term, and sometimes I have to say a bit oversold. What biomarker currently in kidney cancer is something that you can measure mostly by looking at the tumor or the blood that can alter your management. So perhaps, there is something where you don't need to do surgery, or you need to do surgery, extensive surgery. When it's treatment and the patient, is not a surgical candidate, you choose treatment A versus B. And as of today, January 14, 2021, COVID times, we don't have a biomarker. You and I, Bob, when we go to clinic on Monday, we're gonna use. We don't. We have some very nice curve. We have some association. We have some interesting, thoughts, but we do not have biomarker. I think when I think about something that will change my management, I think about screening for kidney cancer before kidney cancer is there or is very, very small. Second is when Dr. Uzzo removed the tumor, should I give a treatment, yes or no? That's a biomarker. And third, if the tumor can be removed and need to be treated by what we call a systemic treatment, something IV or oral, what kind of treatment I can do? Because I don't have one treatment in kidney cancer. And a biomarker could be from the tissue, from the kidney that was removed, whether it was removed or you need a new biopsies, could be from the blood or could be from the urine. When we talk about screening or detecting a recurrence, the cancer coming back before the scans, can show us or whatever imaging, we try as much as possible to do the least invasive thing. So that's where blood and urine come. And one of the, research we had was my collaborator, Matt Friedman, as well as our collaborator in the University of Toronto. We looked at blood and urine in earliest stage patient. And kidney cancer was one of those tumors that was hard to look at. And we found that we could predict over 95% of tumor that have stage 1 and stage 2 disease. But that's not, we looked at a different way. We did not look at the mutations of these tumors, of this cancerous DNA floating in the blood, because if I take blood in you now, there is DNA in it, over 98%, even if you have cancer that is spread, is going to be normal DNA from cells, normal cells that are hanging around, breaking and getting their DNA. So you have to get that tumoral DNA. And then if you have an earlier stage 1, you bet, it's not 2% tumoral, is way less. So now we are talking technology. So one way where we went around that is rather than looking at the mutation that could be present, these alteration in DNA in other tumors, we looked at the methylation pattern, which is give you a bigger idea of the whole pictures and pattern that you can compare and use what we call computational biology and big data set and try to look at it differently. And we did it from urine, because one of the things that, big institution, and I reach out actually to Rob, he knows that he has one of the largest biosamples, not that he did, all the time he had questions, because he collected them of urine and blood in the country, where maybe we'll do it in urine. If you think blood is invasive, urine is, I would say, useless. You want to keep your blood sometimes, but your urine, I think you can shed a lot every day. So we're trying to go that way. And that is the way of screening. So one day my dream, and one of our colleagues that does bladder cancer, a tumor that we all work on, showed it recently, is that if a patient comes and Dr. Uzzo does surgery and they have stage 3 disease and they was a high grade, and we code them a number based on Dr. Uzzo and his team work with these very elegant nomogram. When they put the stage, the grade, and multiple other things, I usually go on his website and, to help. But we forget that there's a subset of patients that are cured with surgery only. Scans can't tell that till the cancer has spread. So could we detect cancer in the blood, knowing kidney cancer is different than lung cancer? It doesn't shed. Well, some of the evidence it's coming that we could do that. And with one of our collaborators, we were able to do that in brain cancer, not kidney cancer, in glioblastoma, which is a tumor that is the only tumor perhaps that shed less DNA in the circulation, because it's all here, than kidney cancer. And we don't know why kidney cancer doesn't shed DNA. It's an area that is very vascularized. So I think that's when you talk biomarker. Now you come to metastatic disease and you have a treatment. The treatment that Dr. Uzzo talked about is a PD-L1 inhibitor and a VEGF inhibitor, atezolizumab and bevacizumab. An elegant work from Dr. Moser, Sloan Kettering, and Dr. Rini. Also Cleveland Clinic, Cleveland Unite Us All, but now at Vanderbilt in the South, showed that there is some patterns of tumor changes at the DNA level, at the RNA level, that predict which patient can benefit from the drugs used on that trial. Well, one of the problems we are aware is these two treatments are less and less used. So what do we do? And how do we validate that if the treatment, if the field is advancing very, very, very fast? We did our own work with immunotherapy, looking also from clinical trial, Rob, with nivolumab, all the studies, and we actually didn't come up with anything. And that's fine when you come up with negative. We didn't come up with anything, but we understood, for example, a lot of things. We understood that one way to look at things is that maybe we shouldn't be driving the immune cell inside the tumor. In bladder cancer, you should, because they're sitting and waiting. In kidney cancer, over 70% of the tumor have already the immune cell, the T cell there. They're just not functional. So it's a different way to think about it. And we discovered also that when you have those immune cells in the kidney, it doesn't mean, you will do better. So, I mean, we have a lot of clues, why things are, a bit different in kidney cancer. It doesn't mean always that we're gonna immediately have the answer, but we're putting pieces of the puzzle, together here. We found, for example, that these tumors that are infiltrated may not be rich in this mutation in PBRM1, may be rich in other, bad mutations. So now we have this interplay between the immunohistochemistry and the, immune cell and the genomic alteration. So those are one of the challenges we face every day. And the other big challenges is we are pushing the envelope, meaning, one drug rarely, we are putting two drugs, and there are now trials that putting a third drug. Now that, don't get me wrong, the toxicities, could be higher, So it's done in specific patient at higher risk. But if we are reaching a response rate that is that high, how can we find a biomarker here if your response rate is really that high? There are examples. Testicular cancer is cured with 3 chemotherapy drug, BEP. There is no biomarker for that. You come, you read, this is germ cell tumor, testicular cancer. It's metastatic, it's spread. You still can cure it when you give 3 drugs together. You give 1 drug, no. 3 drugs together, and it happens. So despite that I do research in biomarker, I want to find at the end of the day, the right treatment while testing in real time, the biomarker. One thing I also want to add is that when we look at genomic information at the DNA level, at the RNA level, we take this piece of the tumor and we grind it, all together, and we come up. So imagine if you have a drink, a smoothie, where you put all things together compared to a small basket or fruit. What we're trying to do now with some clues is looking at the single cell level, knowing it's not just 1 cell. So when we take that specimen in the OR and that specimen is still fresh, didn't get altered with whatever you add, you put it on cold, the tumor is still, if you want, living, if we can use that term. We are trying to take it a piece apart, break it in compartment, and look at the expression of each gene, the expression of genes in each group of cells. And we're discovering so many things. So for example, in one of the studies that Dr. Uzzo mentioned, we stained the tumor with the T cell, with the CD8 T cell. But when you look at the single cell level, the CD8 T cell comes in so many flavors, some of them good, some of them bad, some of them we don't know what to do. So yes, it's more question, but these technologies were not present before, and they were present. If they were present, they were extremely expensive to do and conduct. Now they're becoming mainstream. This is why I remain very hopeful that something gonna happen, even by luck. We'll stumble on something. I'll take luck, every day.
So let me jump in here. We've got time for, I think, one more question, and I'm going to sort of 2-prong it here for you. Dr. Uzzo, when you've experienced the last 10 months, 12 months of this pandemic, can you talk about any silver linings that have come out of that in terms of, your care? And then I heard you say and talk about in the future holding artificial intelligence is going to play a bigger role with neural networks. Can you touch on that just a little bit more so we can get a little flavor where we might be headed?
Let me start with that one first. So clearly, big data is, not something that, is unfamiliar to most people here. The better the processing power of our computers, the more we can aggregate the data. And I see in the chat session, someone asks about, machine learning, and it requires people to be willing to share the data. So you don't only have to have the infrastructure, the technology to do it, you've got to have the infrastructure to do it. And increasingly open source types of data codes for randomized clinical trials is going to be requisite. Now, obviously, pharmaceutical companies and institutions put millions of dollars into these data sets. And one really good example of how this is working well and how the market's allowing it to work well is something called Flatiron. So Flatiron is a company that was founded by a couple of Google, insiders that really has come up with this model where they go to cancer centers and large hospitals and they say, why don't you share your data with us? In response, we will clean the data, we will standardize the data, we will allow everybody's data to be measurable, using the same sort of, metrics. And then you'll be able to peer inside that data. You'll be able to look how your data compares to other people's data. You'll be able to do research on the data in aggregate. And then we'll have a data set that we can then start to, use for real world data to show, what kind of responses people are getting, not in one institution, but in multiple institutions. That's a great example of how, big data is getting used. Another great example is these new foundation medicine type of companies that you can send a tumor outside of your institution. They'll sequence the tumor. They'll do what's called whole genome sequencing. They'll send you the sequence. And so now you've got the sequence and you can attach it to the patient's prognosis and what happened to the patient, et cetera. And so pharmaceutical companies like Hoffman LaRoche are buying these types of companies so that they now have the connection between biomarkers and genomics and outcomes, and they can start to make these connections. Now, is there going to be some sort of siloing? Of course, it's the market. Someone's going to silo, right? You know, you're going to have to break down the barriers to that. But I think that they're coming down faster than they ever have been. So that's the answer to your first question.
Can I add something to that?
Sure.
You know, one of my heroes actually was at Cleveland Clinic. Rob, you probably know him, and he consulted you, Eric Topol.
Yes.
You know, that is now, I would say, making an amazing, reinventing himself with artificial intelligence and social justice. You know, a couple of his books. So, you know, he gives a lot of example of artificial intelligence applied to medicine, applied to life in general. The one that stuck me the most, and of course, I am not an expert in that. I barely know how to type and turn on a computer, is an experiment that they've done where they looked at thousands of ophthalmologists. You know, they looked at the fundus exam. So how you look, you know, when your ophthalmologist look like this, and they fed the computer thousands of those images, and the computer, you know, whatever, you know, I still call it the computer, was able to detect extremely closely who is a male and who's a female. So there is no way, when they compare them to experienced ophthalmologists, there is no way they were able to get, you know, they were like 50-50, a toss of a coin. So what did, you know, the computer, what did they picked on when you feed them every, you know, all that artificial intelligence that were able to answer this is something hopefully we can use in cancer, in kidney cancer. I think that's the most striking example, but you have to remember, these are not, I would say, there's a lot of things that they picked on that surprise you, that it's so silly. It's not like that. Another example where the need, so I want to get a bit outside kidney cancer, very, very large hospitals in India where patient, you know, you have thousands of patients coming to the ER sometimes, and you have 1 to 2 radiologists, and you're looking at chest x-ray, you know, with fatigue, if you didn't have enough coffee, whatever, I mean, you may miss something. So for normal, you know, chest x-ray, this can go to a specific algorithm, you know, that can sort it out. And wherever something, especially for young people, non-smokers, something is abnormal, that get elevated to the radiologist, and you can cut off 90% of those exams. That's one of the way. How is that going to apply to kidney cancer? I think it's coming. I think, for example, you're going to have millions of scans from Dr. Uzzo's practice in Philadelphia linked to tissue, you know, linked to pathology, clear cell RCC, benign tumor, fibro, you know, cyst, chromophobe RCC, and hopefully by some sort of neural network, I do not understand. I even don't know if I'm pronouncing the right, you know, terminology. You will be able, by a specific CT scan or MRI, to know at the 95 or 98%, you know, what that is. So you could observe, you could do a biopsy, you can go directly towards surgery in the right context. So if this is 95% benign, not 100%, in an 80-year-old gentleman that has medical condition, that's different than 95% benign in a 40-year-old marathon runner. So you still need the physicians. Rob, we're still going to have a job, hopefully. Okay, we have young kids. So that's how I look at it.
All right, thank you, Toni. I got to keep us back on track here. Before we turn to the questions out there, we're going to ask a couple of polling questions. I hope I'm going to get this correct here. These these polling questions will be anonymous. We're not going to share anybody's name or data. If we can try to pull that up, I'm not sure if I'm going to get to see it or not, but if the polling question could be pulled up, there we go. We'll take about 20 or 30 seconds here. So what brought you here today? You can answer as many times, as many as that apply. And then when you finish that, if you could let us know a little bit more about yourself or about these items that you're interested in. Great. I see the bar graphs are moving here. Seem to be making some progress. All right. I don't know how much time we need, but let's, we're going to end the polling in just a moment. I think while we're ending up on the polling, I'd like to ask for we're going to turn it over to hear our first question. This is from John down in Texas. John, you want to ask your first question?
Yeah, hi, how y'all doing? Thank you, Brian and KidneyCAN for putting this town hall on, and thank you, Dr. Uzzo and Dr. Choueiri for your time today. 11-year kidney cancer stage 4 survivor, so very interested in everything that you're talking about. And my question specifically is, what are your thoughts on the COVID vaccine, and should patients, whether on treatment or off treatment, receive the vaccine?
I mean, so I've been doing actually some COVID research with a lot of my fellows. One of the things Rob and I enjoy a lot is mentoring young people. It makes us feel young. That's the only, maybe the reason. So we are part of a large registry of cancer patients who got COVID. I mean, so far, very few got a vaccine. And I would say the general answer is yes. I mean, I could go into details why is yes. I am actually, after this, after this talk, I'm taping a talk to the American Association of Cancer Research, where I was asked to talk about systemic therapies, so chemotherapy, immune checkpoint inhibitor, target therapy, and cancer in general in COVID patients. So you could think about the vaccine as COVID. COVID, you got the virus, you mounted an immune response. The vaccine, you know, is another way to mount a similar immune response that protect you. They're a bit different. One is exogenous. You're getting, you know, not the virus. The other one, you're getting the virus. But if you look at COVID patient, we have more and more data that, for example, immune checkpoint inhibitor are safe. Chemotherapy in general, they tend to be safe. Some of the data says that within 2 weeks, there is potentially an increase, you know, of mortality, but that's in a setting where a patient also have other things. So I would say, you know, they are safe. And I would say if you have the chance to do it, have it. We have data potentially from some specific therapies that bring the B cells. That's one of the immune, you know, when you talk immune system, you know, you talk T cell, B cell, the B cell that produce antibodies. Some of the treatment in cancer bring them down, but it doesn't mean there'll be harm when you get a vaccine that is not, you know, a live vaccine. You know, and the 2 vaccines available today are RNA vaccine. They're not live vaccines.
So Dr. Uzzo, real fast, we got 20 plus questions here. Do you have anything to add to that?
I would just agree. I think they're safe. Remember, here's the order, John. DNA makes RNA, RNA makes protein. That's the order, all right? So people worry about RNA getting into your DNA. It doesn't go backwards, all right?
Yeah.
DNA makes RNA, RNA makes protein. So they give you RNA, it makes the viral protein, you make an immune response. You know, all the data suggests right now that should be safe. I took the vaccine.
Great. Thank you.
Thank you, John. Appreciate your question. I think we're going to try to move on to the second question on our list. I don't see them popping up right now. Oh, no, I think I might see the name popping up. Cindy, or can you come up live? Okay, maybe we can just hear from you then. I think your screen's turned off, Cindy. But if not.
Cindy,
Okay.
if you can hear me, you should have a button in your bottom left-hand corner that's a microphone. If you go ahead and hit that. If not, Brian, could you ask a question on Cindy's behalf, please?
So Cindy's trying to get to the button, but maybe not. Okay, well, I'm going to ask the question if she comes on. In the interim, are there any non-TKI treatments in the pipeline that may become available in the near future?
Well, Rob, I feel I talked a lot.
Well, I will answer the question. Yes, in generally speaking, you've got TKIs and you've got immunotherapies. Those are the two big categories currently. But then, as I mentioned before, there are new things where there are metabolomics that are in the pipeline. And then the big one, as I think we'll get to, are the Hif inhibitors, the hypoxia-inducible factor inhibitors. Just like I was saying before, the VHL protein gene makes VHL RNA, makes VHL protein, but that protein then regulates other proteins. So there's a whole pathway, kind of think of like steps on a ladder. And as you go up the ladder, you can get more and more and more specific. So, we went from the TKIs, which actually control sort of, the receptor, to now to these Hif inhibitors, and the phase 3 data looks really good on that. I think that's the next thing that'll be approved. Tony?
Yeah, I agree. I mean, I put these under small molecules. Again, this was a win. These Hif inhibitor went to science because transcription, factors were hard to target. A lot of these protein molecule, they have friendly pocket where you can design or, come up with a screen that get a drug inside the pocket. The transcription factor don't have, these friendly pocket. They do their job in a very, interesting way, how they interact with protein and the DNA. But, more hard work and led to a first generation molecule. Then a second generation molecule that in a phase 3 trial. The interesting thing about these Hif inhibitor, and as a conflict of interest, I have to declare I'm involved mostly in a leadership or co-leadership with probably all these trials. So I want to mention that the interesting thing about them is that the toxicity is a bit different than these TKIs. When the toxicity are different, you can combine. When we think about maximizing therapies, we think about, in a way, synergy was where 1 + 1 = 3. Although there's a lot of data coming that synergy may be oversold. And at the end of the day, you need a drug that works one way and attack parts of kidney cancer through a mechanism of action. Another drug that also as a single agent attack kidney cancer cell and kills them in a totally different mechanism. And the side effect profile of both is totally different. So you can combine them. Imagine if both of them, different mechanism of action, but they have high blood pressure or dropping the counts together. So you cannot reach higher level. With these, the HIF2 inhibitor and TKI, the side effect profile is different. So we are starting to combine these drugs together, and hopefully, they will be better than one, and the rate of responses will be higher and higher.
Great, thank you very much. Deborah, coming from Massachusetts, can you come on up and ask your question?
Hello there.
Hello.
Thanks for being here. And I'm a 4-year advanced kidney cancer patient. And what I'm interested in is hearing more about the HIF2A inhibitor, the MK6482. In specific, my question is, what is the status of the clinical trial for advanced RCC? And when might the approval be? Is there any estimate for the FDA approval?
I wish I can put the hat of the FDA and decide. What we know now, Deborah, is there's an ongoing study in patients whose tumor has grown on immunotherapy and on TKI, where they get the control mTOR inhibitor, everolimus, versus the HIF2 inhibitor. And we have it open at Dana-Farber, and it's open in many, many places. But also what we know, there is a specific patient population that is called, VHL, which is a small but very important population of patients that taught us a lot about renal cell cancer. These patients have mutations, inherited mutation in the VHL gene, for which there's a study that showed an impressive activity there and low toxicity. So it is possible, and usually with these rare tumors, sometimes you don't need this very large studies, phase 3. It is possible that an approval will come for this population, and the drug will be approved. Now, could we use it in kidney cancer? How? That's gonna depend on many factors.
Great, great. Thank you, Deborah. We got 16 or 17 more to go here. So can we bring up Sherry from Nebraska? Sherry, I think you're turning, there you go. Hi, Sherry.
Hi. My question is along the same line of the Hif inhibitor. And what are your thoughts around the possibility of this being used in later lines of therapy?
I'll start to answer that question. So the way clinical trials almost always work and approvals almost always work is you work from the most advanced people who've tried, most everything that's already approved, and you start working it backwards. And as Dr. Choueiri mentioned, in the past, we used to do clinical trials comparing new drug X to placebo, but then we realized you can't do that anymore because TKIs work. So then it was new drug X to TKIs. And then you started saying, well, these combinations work. So it's combination A to combination B. And so you start to just sort of make these iterations. So what the iteration is right now with these Hif inhibitors is, as he's mentioned, Hif inhibitor initially versus TKI, Hif inhibitor, initially versus other immunotherapies. And now after after failure of those therapies, and now it is in combination. So I think that this is just an evolution, and the next logical evolution is to sort of get this thing approved in patients that have failed standard or approved therapies. And then what happens is, hopefully after the FDA approves it, we start to get some real world experience so that people can decide really if it belongs last in the sequence or if it belongs earlier in the sequence. That's just sort of what happens in the real world. And I think that's what's going to happen here. Tony?
Great, thank you. Thanks, Sherry. I got to move on here. We're going to shift gears to getting the best of care theme. And we've got Elizabeth from Florida who's going to come up here, going with Betsy, I guess is the screen name. Hi, Betsy. Elizabeth.
Hi, how are you? I'm in Florida, and I was diagnosed with kidney cancer 2.5 years ago. I've been through several treatments already, and my question is, what is an efficient way to find a clinical trial that I can qualify for?
Betsy, this is an excellent question. There are things, online. Usually the trials all have to be registered with the NCI on cancer.gov. So you can find them, but I find them even myself sometimes hard to read. Even though the status is updated, what do they mean? I think your physician, is usually, it's hard to say if they're general oncologist or oncologist in GU tumors should be able to help. There is a lot of, advocacy group and association that know well, the doctors and the resources. KidneyCAN here with Brian and Susan, everyone is one of them. Also, there are others too. I think that's the way, but start with, your physicians. I would say one thing though, despite being clinical trials, if whatever you're taking now is working, that's great. It doesn't mean you need to change and go to a clinical trial because it sounds interesting.
Yes, I know that.
I still have patients on treatment I wouldn't do today, such as sunitinib, pazopanib, that are doing very well. They were among the lucky folks over 5-10 years. And you know what? This works great.
Right. I know that. Thank you.
Great.
I would just add clinicaltrials.gov, like he said, clinicaltrials.gov, and then go to individual cancer centers' websites. They'll often list their clinical trials on their websites, and you know what the big ones are in kidney cancer, and just sort of periodically scan through them.
Great.
Thank you.
Thank you, Betsy. Thank you. Appreciate the question. I'm going to read in a question that we got from Colorado from, looks like from Ann P. Ann says, I have a local general oncologist and a consulting oncologist at a GU clinic in an academic hospital, but there are no RCC specialists. Do any of the RCC specialists do telemedicine consults and support? I guess that's a really broad question for you all. Maybe you can't answer specifics, but can you give any information on that one? I'm taking that as.
I'll take it. I think that most of us are getting pretty used to televisits and telehealth visits and video visits. And so for consultations, we do them. The problem is delivering care virtually. That's hard, right? So you might be able to get an opinion, and they might be say, yeah, that sounds like it's a reasonable course. Or they might say, well, if you were here, I'd offer you X, Y, or Z, you know, but receiving treatment virtually is difficult. I mean, there's just too much that's involved with getting that care and sort of sharing that care. I think we're not yet in medicine at the point where, you know, physicians can share care across, you know, long distances. Tony?
Yeah, no, I agree. I also found, I mean, the kidney cancer community and the GU, you know, oncology community in general, to be very friendly. And you and I, I'm sure we get emails from all over. You know, people to be medical oncologists, usually they have to go through some intense training or urologist, you know, and, you know, we'll be happy to help. Again, the problem will be delivering, you know, the care locally while we are, you know, far away. And it's not just the doc. I want to mention, you know, also, you know, my success, you know, in delivering the best care for patient is the success of the team. So we have some of the best nurses. We learn from them every day. Today, my nurse texted me about something I wasn't realizing. I had to look it up. And as usual, she was right. You know, the research team also involves clinical research coordinators. You know, we have program nurses. We have folks that do the submission to the IRB. It's really, really a huge team effort that we need to standardize. And that's sometimes, you know, a bit hard to have locally or transfer, but we do our best.
Great. Thank you. I'm going to bring up Karen from Minnesota. Karen, you got a question for Dr. Uzzo, Dr. Choueiri?
Yeah. Often I'm told to find a renal cancer expert, but how do I determine if an oncologist is an actual expert?
If they lost their hair.
That's the best metric. That's right.
That's the best metric. No, you know, I'll take this. I mean, I think with oncology or with urologic oncology, because, you know, Dr. Uzzo deals with cancer. It just, you know, also does surgeries. I think a lot of it is a partnership. This is not black or white or going through your accountant, yes or no. You do have to be very comfortable with that person. And I think, you know, it comes, you know, some research about them, the team. I would emphasize, you know, also on the team, the strength of the team. I mean, if you have a doctor on their own doing everything without the help of all the support staff, you know, everything, the experience, at least on my center, and my former CEO and my current CEO said the same thing. The experience at our cancer center start with, you know, with the valet parking, you know, and the cafeteria when you get your car. I mean, this is all part of it, and it's not really black or white.
Great. Well, thank you for that question, Karen. We're going to try another poll here. So if you're at home, don't leave, but here comes a poll. Hopefully it's a fairly quick one. Would you consider a clinical trial? Yes, maybe, I'd like to learn more, or no. And if we had the Jeopardy music, I would sing it, but I shouldn't, so I won't. All right, looks like we're getting a lot of answers. Interesting, pretty neat. 71% on the yes, 25% on the maybes, 3% on the no's. Where's Deb Maskins when we need her to shout this from the rooftops? All right, so let's shift gears again here. We're going to move into early detection. I think we've got from the great state of Maine, Carolyn, are you there?
Yes, hello. Yes, I'm here. I'm excited to see that Tony's got his Maine lobster tie on. Thank you, Tony. But my question is, where are we with early detection of kidney cancer, and where are we with understanding causation and then with early detection? You have touched on that a little bit, Toni, but could you elaborate, please?
Yeah, no, thank you for the question. I think we're seeing things that we haven't seen before, and that's the ability to be able to detect cancer, kidney cancer, in very, very small amount of blood and occasionally urine. So the things are going. We don't have the same samples as the clinical trial Dr. Uzzo mentioned, but it is ongoing. Today we received, we're part of a collaboration with the French, that collect, usually in Europe, they have collected that type of, data to be able to do something like that. So at some time it is coming, although I would say that imagine how much time it took for larger studies to get a benefit from low dose CT in lung cancer, which is way more common. They detected the smokers, etc. There has been studies, in the UK and many other places, but mostly in the past, we're looking at, ultrasound and, low dose imaging, where they found they'll end up with way more false, positives here. So we have to be very careful, but I think things are approaching, not at the fast rate I want them to be, but I look, when Rob and I were in Cleveland, I mean, it was like, to talk about detection kidney cancer, you will be slammed in a conference. They'll make fun of you. And now we're talking about it openly.
Great. Thank you, Carolyn. Hey, I'm a Red Sox fan, but I'm going to let this New Yorker on the line, our good partner from the National Kidney Foundation. Liz, you want to ask a question?
Yep. Can you hear me?
Yeah.
Oh, great.
You're live.
Hi, I'm Liz Leff. I am Corporate Relations Director at the National Kidney Foundation, as Brian said. I had stage 1 kidney cancer about 6 years ago. They found it incidentally. I had a partial nephrectomy, so I was extremely lucky. I think you guys alluded to this earlier on in the presentation, but I do have a 13-year-old daughter. My kidney cancer is pretty much hereditary or genetic. So I guess I would ask, is there any sort of genetic testing or biomarkers that you could identify earlier on, as well as she's 13, when's the best time for her to be checked out or looked at?
I could take that one, Liz. I'm glad you're doing so well.
Thank
Number
you.
one, every cancer is genetic, meaning that every cancer is caused by a mutation in a gene. That's different than hereditary, and it's common for patients sort of conflate them two. Hereditary means my mom gave it to me, or my dad gave it to me, or I could pass to my kids, right? That's a hereditary thing. So we make a distinction between germline mutations, meaning that you can pass it down, versus mutations in, we call them somatic, meaning that they're mutations in the cancer cells themselves, but not in any other cells in the body. So this is really a function of where's the mutation? Is it in all of your cells, or did it only occur in the cells that became malignant? So if it only occurred in the cells that became malignant, you're not going to pass that down to your daughter, all right? If it's in all of your cells, a germline mutation, then she's at risk. Now, you could be tested for germline mutations, much like Angelina Jolie was tested for BRCA1. You could get tested for a panel. Usually about 50 genes get tested. It's either a blood test or a saliva test. And they could test you, and they could see if you've got one of these germline mutations. In general, we say that if you're younger than 45, then you should get tested when you've gotten your kidney cancer, because you could have a germline mutation. That's a general guideline. Also, if you've got multiple tumors in multiple locations, both kidneys, or if you've got any other symptoms or findings that could be associated with what we call a syndromic kidney cancer. If you don't have that, if you were older than 45 when you were diagnosed, if you don't have any other findings consistent with hereditary kidney cancer, we generally don't check you unless you really want us to, and then you could check. All right, does that help?
That's great. I was 43, just so you know, so I
Yeah, so then getting tested, getting a panel, getting a genetic counselor, they'll test you with a panel, there's a kidney cancer panel. They could let you know if your daughter's at risk.
Thank you so much. Appreciate it.
Thanks, Liz.
Thank you.
Good timing here for some of those people who are on the chat. We are going to shift to the rare subtypes for a moment. I'm just going to ask this sort of general question. What's new for rare subtypes in treatments or trials or care? Let's throw it out there.
Well, kidney cancer, and I think someone gave me this reference, although I don't find it to be tasty. It's like ice cream. It comes with multiple flavor. And 80–90% of people like vanilla, and the rest, like me, I like strawberry and mango and all those fruits. So there are tumors that are genetically, genomically, histologically, when you look at the microscope, different than what Dr. Uzzo and I discussed, the clear cell, the one driven by VHL, and you name it. Papillary, chromophobe, translocation, which we are recognizing more and more, and when we can't tell, it's too undifferentiated and we want to sound smart, we call it unclassified because we just can't say it. Some of them also are very aggressive. They're collecting duct or medullary, which, have some specifics, and they respond even to chemotherapy, cytotoxic chemotherapy, which we use, in lung cancer, and kidney cancer in general doesn't respond to that. And now what we're doing is we are applying the most, I would say, the most, advanced technology, like the single cell we discussed, to dissect these tumors. So we have projects in chromophobe kidney cancer with some of the best scientists, actually Dr. Lisa Hensky that we stole from Fox Chase, right? And she's working with us on a rare tumor that what you do, you throw everything at it, chromophobe. We have also a project with translocation, kidney cancer, trying to understand this very important tumor that we recognize it in young adults. So far, we know it's driven by the translocation, which is not helpful. But if we find another surface marker that we could target, we'll make a drug at some point. But all it's coming, and it's going to take time. But at least there are efforts. And in the kidney, in the KCRS in 2019, in the KidneyCAN meeting, Dr. Uzzo is part of that paper. A rare type of kidney cancer was identified as one of the 6 priorities, in addition to biomarkers.
Great.
If I could just add to that for a moment. So one thing you should know is that the pathologists have classified 15 or 16 different types of kidney cancers. However, within these, there's something called sarcomatoid findings. If you've got a sarcomatoid finding, that's not a unique type of kidney cancer. That's just a characteristic. And in those patients, they really don't respond very well to TKIs. They should get checkpoint inhibitors. Is that right, Toni?
Correct. Actually, I'm glad, it's amazing because that's the tumor I published the most about. We have a paper just, I think, was accepted. It's sarcomatoid, which is one of the things that exists with every tumor. Sometimes it's 100%. And that type of differentiation doesn't respond well to all the TKIs or even chemotherapy. But for some reason, possibly due to the fact that these have a very immune-rich milieu, an increase in PD-L1 expression, many other things, they respond to immunotherapy very well. We have seen responses, a complete response rate of 19% with a combination of nivolumab and ipilimumab to immunotherapy. So I think there are hope for these patients. We're also trying with one of our collaborators, Dr. Van Allen, to dissect this. Actually, we have also one of the things about biomarker I wanted to add is the importance sometimes of biopsying the tumors. It's very hard to ask the patient. It's really research only along the journey. So we have a project actually with Dr. Klimak, who is Dr. Uzzo's counterpart in medical oncology in GU, where we have tumors at baseline when the tumor respond and when the tumor stop responding, because some of the resistance happen and it's acquired. It's with time. It's not at baseline. So we have a project that got a bit derailed because of COVID, but now it's back with Dr. Plemek, Van Allen, Zibelman, and the Broad Institute that hopefully will shed some light into what happens and why folks that respond initially and we clap on then lose that response with time. Some.
I want to jump ahead because we're running low on time here a little bit. We've got some questions on immunotherapy, but I'm going to jump ahead. We've got Kevin who's on the line here. Kevin's calling in from Massachusetts as well. So Kevin, you want to come up and ask your question?
Yeah, Dr. C, Dr. Uzzo, obviously, thank you guys so much for spending time. Actually, I'm on Cosmic 313, Dr. C. I'm being treated over at your competitors at MGH and Dr. Lee. My question kind of is about the timing of conducting the nephrectomy. I think in 2019, it kind of changed. Obviously, I'm getting a great response with the treatment that I'm on right now, and I'm coming to you from my home gym here. But can you expand a little bit on the timing of conducting that nephrectomy? I've got a huge, great response. Almost all my metastasis is gone. Just a couple little kind of dots in my liver right now are left, but my kidney has kind of stopped shrinking. And I know some other folks, it's kind of like, oh, they leave it in, they don't leave it in, they take it out, they take it out. And it seems like it's kind of, nobody really knows what the right answer is. And hopefully maybe you guys can expand on, maybe it's a moving goalpost and we're hoping that, you know, just drag out the treatment and we'll know more information in 18 months and we'll go from there. But so that's my question.
I think it's a great question. I think what you're asking, just for context, is if somebody presents with stage 4 kidney cancer and they've never had surgery and their kidney's still in place. In the past, we used to remove the kidney and then give them some therapy. And a recent study suggested that probably what you ought to do in most cases is give them the therapy before you remove the kidney. And so what happens is patients like you, they get the therapy and they've got a great response. And the question then becomes like, you know, should we stop the therapy to take out the kidney or should we keep giving the therapy? So in general, you know, that question's not really been fully answered, but the practice is give the therapy as long as you can and the patient's tolerating it well to get the maximum response. Once you believe you've gotten the maximum response, then usually we go to our tumor board, which has the surgeons and the medical oncologist there, and they sort of come to a conclusion. Can we safely get this person through surgery quickly and get them back onto therapy? Because unfortunately, we sometimes see if you go to surgery and take out the tumor and you get like a 6-week or a 12-week break, then suddenly things start to activate again, right? So you don't want to put a person that's doing really well at risk where you open up a window that suddenly things turn back on. So the question really, Kevin, that you're asking, that you'd like answered is, once I've maximized my therapy, can the surgery be done safely and quickly and get me back onto therapy, you know, if necessary, so that I don't have this big gap in my coverage, if you will, Tony?
Perfect. I mean, it is, you know, we think about medicine. Medicine is, you know, half science, half art, and this is art. And Dr. Lee is a, you know, I don't think of him as competitor. He's a very fine man, also from Cleveland, by the way. So we're all here, you know, have a relationship to Cleveland. And we have that scenario very common, independent of the therapy even.
Great. Kevin, thank you very much for that question. We got a few more we're trying to squeeze in here. We've got Brian from New York. I hope he's out there standing by. Brian, can you come up on screen? You might have to unmute yourself. You've got to unmute yourself, Brian. There you go.
I thought
You're live.
Okay, can you hear me though?
We can.
Okay, I have stage 4 clear cell, and I'm being treated with the Benevo up in the Wilmot Cancer Center in Rochester. And the only issue right now is I think I need a new oncologist because mine has a full set of hair. But anyway, my question is, how long should one stay on immunotherapy if one is fortunate enough to achieve a complete response?
You know, I mean, this is the question we're trying always to answer. I think if people have a very good complete response, or what we call a deep response, you know, even from kidney cancer expert, I heard anywhere between 6 months to never stop if it's tolerated. In my practice, it's a discussion with the patient. If we have a near complete response after a year or 2 years, especially if side effects start to happen, a colitis, you know, a hepatitis, thing like that, I stop and hold and restart if needed. We had some patient having what we call immune-related adverse event and toxicities after 6 months, 12 months, or 2 to 3 years out of the blue, which we never thought, you know, would happen. So that's in a way. So deep response, in my book, it's usually 2 years, but we've seen things as short as 6 months when we had to stop the immunotherapy because of toxicity.
2 years after a complete response?
No, sometimes 2 years at the complete response, because complete response usually preceded by several months or a year of near complete response, or what we call deep partial responses. Usually that's the pattern.
And I'm thinking, based on the discussion, these biomarkers in the future may help make that determination.
I mean, we hope. One of the projects we've been involved with is to look in these patients at the tumoral DNA in the blood. So imagine if we can't detect it anymore and it was detectable before and the scan are there, perhaps that's a sign. That's something we have to do.
Thank you.
Thank you, Brian. We've got folks that are still hanging in there with us. We got 3 more questions. So if you can bear with us, we'll get to those. It looks like we've got Tracy coming up from Ohio.
Ohio, yeah.
Sorry about your Buckeyes.
She can have half an hour.
Hi, everybody. I'm actually a caretaker for a patient being seen at the Cleveland Clinic. So we got to get another shout out to the great team up there. My question for you is, I'd like to hear what your current thinking is regarding the best second line treatment after the Pembro-Axi combo has stopped working. Thank you.
Thanks, Tracy.
Yes, I do not want to make, you know, general statement, because I have done almost everything after Pembro-Axi or VEGF-IO, including surgeries in some patients that had progression in one place, or radiation, or treatment A because they couldn't, you know, tolerate treatment B. You know, I have used cabozantinib, which has data post-IO. I have used lenvatinib and everolimus. It's very, you will be surprised how much it is context dependent when we dissect the chart. But the goal here for you, Tracy, is that there are options and there are more than one option.
Dr. Uzzo, you want to drop in on that, or is that covered pretty well?
I think that's covered well.
All right, then thank you very much, Tracy. Go Browns, I guess I should say. I've got a good friend who's here in Pennsylvania that might be coming up next. He's one of our veteran advocates on Capitol Hill from a few years ago. Michael, are you standing by to ask your question? I think you got to turn your camera on. Might be coming. Nope. Yeah, you're almost there. You might be on mute as well, but give you another second. Oh, I think you're off mute. Michael, you there?
Yep. Can you hear me now?
We can, but we can't see you, but we can hear you. So speak.
Well, that's probably better off.
Okay.
Thank you, doctors. Thank you, Brian, for this great event. It's really amazing. And I cannot go without saying thank you again to Dr. Uzzo for almost 20 years of survival. So I'm very, very happy with that. My question is, which I've had, is what is the role of Sbrt on RCC Mets? And when is it appropriate?
Thank you, Michael. Let me try to answer that quickly. So in the past, radiation was only used for symptomatic metastases. That is, if they were bleeding or if they were painful, we would use it just sort of to treat the symptom, because it's actually very good at decreasing, growth rates and at decreasing sort of compression of the tumor on normal tissues, which is really what's causing the problem. What we now are doing, though, is because we can deliver a lot higher dose without so much toxicity to the surrounding tissue, we're starting to use Sbrt or targeted radiation therapy, cyberknife, those types of things, to actually treat it for therapy, not just for symptoms. I will tell you that, we generally use it as an adjunct. It's not usually the first therapy. We generally use it more for bone and brain than for liver and lung, because those tissues don't tolerate radiation quite as much. So to answer your question in brief, we use it for symptoms, increasingly for therapy. We use it targeted to certain organs, mostly brain and bone. We don't use it for other organs that don't tolerate radiation quite as well. Tony?
Yeah, I don't have anything to add with that, but this is coming and more data is accumulating. But surgery has the best track record in general, years and years of follow-up.
Okay, great. I think we're down to the 2-minute warning here. Let's see if we can bring up Corey. Corey's down in Florida. Corey, you have a pretty interesting question, it looks like. Oh, you're on mute, Corey. You need to, yeah, you're still on mute, Corey. There you go. You might be live now. Oh, we still lost you.
No, I think we've got a mic issue for Corey. Sorry, Corey. Brian, would you like to go ahead with Corey's question, please?
Sure. Sorry, Corey. We seem to have some audio challenges. Here's the question that Corey sent in. Can we talk about oncolytic viruses and have the trials been effective?
I could only give you a very quick background on that. What Corey's talking about is some viruses actually have been associated with the creation of cancer. Think about cervical cancer and papillary and HPV. Think about HPV and throat and tonsillar cancer. Think about hepatitis and liver cancer. So there are, Burkitt's lymphomas and EBV or Epstein-Barr virus. There is not yet any virus that's been the associated virus with kidney cancer that I'm aware of. That's what Corey's talking about. There are, I think, as we sort of do more and more of genomic sequencing, we might find bits of viral DNA in cancers like kidney cancer, but that cause effect has not yet been established. Tony?
Yeah, I agree. Can you hear me?
Yeah.
No, I totally agree. I think this is a new class of drugs for the treatment of, cancers. So these are viruses engineered to kill cancer cells. We have one approval, to my knowledge, in melanoma with a drug called the TVAC. I would say that this is still really in the domain of research. These are viruses that preferentially, for whatever reason, infect and kill cells. I think the biology overall is extremely fascinating, but the application is still yet to come. But it is coming. It is coming because of multiple, it changes how to deliver, that virus.
So just to be clear, so Tony and I approached that question exactly in the opposite direction. I approached it, can viruses cause kidney cancer? He approached it, can viruses cure kidney cancer? And the answer is probably, to some degree, yes and yes, but we haven't figured it out yet.
Yeah, I agree.
Thank you, Corey. Appreciate it. Sorry about the audio. I think we're on the final two here. Maybe number one, it's on triplets. There's Ipi, Nivo, Cabo. There's Nivo, Nectar, Axi. I can't even keep track of them all. Maybe you all can educate us. I know, Tony, you mentioned triplets earlier as possibly a solution. Any other thoughts?
I mean, again, I think the concept of combination, how can we drive these responses, especially complete responses up? And again, this is how we cured cancers. I mean, if you see how we cured cancers that are advanced in lymphoma and leukemia and testicular cancer, these are advanced cancers that are spread, is by doing combination treatment. So do we dare to dream in kidney cancer? Yes, when drugs are tolerated at the right dose, have alternative mechanism of actions. And we have now trials with 3 drugs versus 2. So let's see how that can pan out, because at that time you need to have some sort of benefit that the community gonna agree. What's the degree of benefit? Is it gonna be a difference in 10% response? Is it worth it when you look at the toxicity? So remain to be seen.
All right, we're in overtime. This is what I think is probably the last question from Deb M up in Canada. Where are we with adjuvant trials in terms of timelines to read out?
This is topic dear to your heart, Rob and I. So Rob, you can talk, I can talk, because we're both, involved with different but same trial.
Well, so kidney cancer adjuvant trials, I alluded to it before, there have been at least 5 in the immunotherapy space looking at single agent immunotherapies and checkpoint inhibitors to decrease the risk of recurrence of kidney cancer. When do we think they're going to be read out? I don't believe before 2022 or 2023.
So the other thing, I agree. So when we say adjuvant, I think the thing is, part of me, yes, we want to cure patient and decrease significantly the risk of recurrence, but we are over-treating. We're going to be over-treating patient because some of the surgeons, the most aggressive pathology report are still cured by Dr. Uzzo and Kutikov and other surgeries, and that's one of the things. I think the approaches are going to differ. Luckily, we have trials where you remove the kidney and you give them pembrolizumab, atezolizumab, that's a trial Dr. Uzzo involved, or nivolumab and ipilimumab, 2 drugs, that's another trial, I think with Dr. Moser and SQDA. But there's a very, very important trial that is going to finish accrual soon called the PROSPER trial, which is open in the United States, in Canada, and I believe in Israel, which is a very interesting trial where you give the drug, the immune checkpoint inhibitor before and after, because there is a chance that there is a lot of, mounting evidence from the lab here that your immune response may be better if you expose your system, if you give the checkpoint inhibitor to more antigen present. And when you have the kidney with everything present is different than you have a couple of cells floating. So that's the PROSPER trial that is extremely important and complementary to the adjuvant studies, for which we have it open here. I think you have it, Rob, open. It's a bit more harder work to do because you have to get the patient, but it is worth it. And I'm very excited about all these trials.
Wonderful, folks. We're 5 minutes in overtime, so I wanted just to say thank you to everybody that stayed on this long. If you are interested in staying around for 30 seconds after we wrap up here, we have another poll. I know some of you have to run off and get dinners ready on the East Coast at least. Toni, thank you so much. We really appreciate everything that you've offered, and Dr. Uzzo, really appreciate you spending time with us. We're getting all kinds of responses on the chat. Really appreciate it. Thank you for taking the time tonight. We really do appreciate all this insight.
Thank you. Stay safe and don't forget the mask.
Great. Thanks, folks. There's one more poll if you're still on. If you do want to share information, this is where we would be needing some more private information, I guess, if you're comfortable, because we're always trying to recruit folks for our Army to go up onto the Hill and do virtual as well as in-person advocacy work for kidney cancer research funding. But I just want to say on behalf of KidneyCAN and the entire community, thanks for joining us tonight. It was really special to hear from these 2 experts. Don't forget to wash your hands, stay healthy, socially distance. We'll see you soon. Thanks so much. Take care.